Well, thanks for coming, everyone. It's my pleasure to introduce Dr. Samay Pande from the Indian Institute of Science in Bangalore, in India. So Samay's research explores microbial predator-prey interactions. So I think many of us tend to think of, microbial systems as being, very flat trophically. But in reality, there's bacteria that consume other bacteria that are predators. And understanding the eco-evolutionary dynamics of these systems is a really big focus of Samay's research group. And then the other thing that I think we tend to forget is that selection for antibiotic resistance has been going on for a lot longer than it's been going on as a consequence of human usage. Obviously, microbes themselves use antibiotics for competition, defense, and predation, and so on. And that's another big focus of Samay's work. And I think what he's going to be talking to us about today is how does predation drive maintenance and evolution of antibiotic resistance. So it's a really exciting system and some really interesting questions. And I'm super excited to hear Samay's talk. So please join me in welcoming Samay. And Samay, please take the floor.
Thank you, Andrew, and thank you everyone involved in this seminar series for this nice opportunity. This was really nice introduction, and I think I should just wrap up and say we can go to the questions because this is exactly what I wanted to preach about. I've been trying to preach about this topic for some time now, and I thought I'll do it again because, reaching out to probably a different set of audience. So if there are people around who are again attending this talk, this might be part of this might be repetition, but there's hopefully some fresh information for all of you in this presentation. So as the title suggests, I'm going to talk about how microbial interactions, specifically predator-prey interactions, influence the maintenance, that is ecology, and emergence, that is evolution of antibiotic resistance in microbial communities. Now, before I sort of jump onto some of these observations, which will hopefully make these statements more clear, add some information and data to these statements, I'll also take help of these series of Hollywood blockbuster movies. I'm a huge fan of those, especially the novel on which the first couple of them were made on. And the reason for this fascination is because in this novel, you sort of get to read, of course, in a very popular language about how dinosaurs sort of evolved mechanisms to hunt efficiently that can be having a stronger jaw, a longer claw, etc. And how different organisms might have lived in the presence of this predator, and that can be, being another dinosaur, which is just too large. So essentially, we are talking about co-evolutionary dynamics between predator and prey. In addition to that, I think it's also very fascinating to sort of kind of imagine or test what happens when you introduce a predator in an environment where rest of the organisms have not seen this predator in their recent evolutionary history. So I'm not going to talk about this second bit, but I just wanted to advertise this bit that we are sort of keen on some of these topics in addition to co-evolutionary dynamics between predator and prey, sort of the effects of dormancy and community assembly, etc., of predation on microbial communities. So this was just to give you sort of a sense of the kind of themes that my lab here at ISC is interested in, and our questions are sort of broadly ranged, sort of based within these few themes. Now, of course, if I want to study predation, and these Jurassic Park is the theme that I wanted to go with, we should have started working with dinosaurs, which we can't. So the simplest thing to do is to study bacteria. And we use two different approaches to study bacterial predation. And when I say bacterial predations, just to make it sort of very clear and distinct, I'm not talking about eukaryotic or multicellular predators that kill other bacteria, but I'm talking about predatory bacteria, which means a bacterium killing another bacteria and eating that dead bacterial cell as their food. So both interacting partners are microbes here and bacteria in these system that we work in. So here are two approaches that we deal with. We study natural, I'll just get the pointer on. Okay. Oops. Can you see my cursor anyways?
Yeah.
Yeah, perfect. Okay, so that's enough. Okay, so we study two approaches. We study natural microbial community, diversity in these communities, and how these predators sort of might be affecting these communities. And we'll talk about that in a bit. And based on our observations studying natural microbial communities, we developed some of our hypotheses. And those hypotheses we sort of study on an evolutionary timescale using lab evolution experiments. And to study bacterial predation, the model bacteria or model predator that we are using is called Myxococcus xanthus. And this is a gram-negative bacterium, and it's a soil microbe. And wherever you are living on this planet and watching this stream from, I can guarantee that if you take 10 different soil samples, let's say a column of 10 centimeter by 5 millimeter, at least four of those soil samples will have this microbes. It's a very widely spread bacteria across the planet. And for a microbe, for a prokaryote, it has a very fascinating life cycle. Of course, they can live as what we call as vegetative cell, just like E. coli, eat food and do binary fission, divide and make copies of yourself. But in addition to that, if you starve these populations of these bacteria, they tend to aggregate together. So hundreds and thousands of these cells will come together. And within these aggregates, the individual cell tries to become a spore. However, only minority of the cells, that is about 1% of the cells or less than that, manage to become a spore. So what is a spore? A spore is, you can think of as a sort of a dormant seed of bacteria, which when conditions become favorable, they can germinate and sort of life cycle goes on. Okay, so when you starve these bacteria, they form these spores as an aggregate within an aggregate, and when conditions become favorable, they germinate and life cycle continues. In addition to this, sort of one of the topics that we are going to highlight today, they are predatory bacteria, which means they can sense other species of microbes in their environment, kill these bacteria, and use these dead cells as their food. Now, interestingly, predation strategy here is commonly group predation. So, of course, individual bacterium can hunt another microbe in this case, but majorly because digestion of a prey, it's not a eukaryote that it can engulf a cell. So, for a digestion of prey, they'll still have to secrete digestive enzyme, and therefore, idea is that multiple predatory bacteria attack a prey to sort of consume it. Okay, now for the purpose of this talk, we will highlight only these two aspects of M. xanthus life cycle. So we are talking about a microbe that can kill other bacteria using basically any antimicrobial strategy that microbe can make. So if you look at the genomes of myxobacteria, every antimicrobial strategy that a bacteria can express, these bacteria have it in the natural isolates. And here's a picture which is sort of showing how this area of a myxobacteria swarm basically invading a circular colony of E. coli, killing these E. coli cell and lysing them. Now, among these antimicrobial strategies, one thing that these bacteria can do is produce antibiotics to kill their prey. Now, the second aspect that I want to highlight is these microbes, as I said, make spores. So they're not going to die anytime soon. If you make them stressed with pH or nutrients, etc., they will just transition themselves into a spore population. And as a microbiologist, what we are very used to seeing, and that this is what also makes it difficult to work with these bacteria, is that these are very sticky microbes. So now we are talking about a microbe that is very sticky and is not dying. So literally on a single physical surface, this microbial population is going to stay for a long, long time. And by long time, I mean in sort of a microbial number of generations, not on a geological time scale. Uh, and this bacterial population that is staying on a given location for a long time can also produce antibiotics. So, sort of bringing all of these aspects of M. xanthus biology together, a couple of students in the lab, essentially Saheli, who was a grad student then, who was also helped by Jyotsna, asked a asked a simple question, that is whether presence or absence of M. xanthus in natural soil communities have any effect on the abundance of resistant bacteria. And for that, what they did is they essentially collected soil samples across our campus. So this is sort of our map. And they collected samples from north, south, east, west regions of the campus. Of course, not only five. The data I'm going to show you is for more than 25 samples. And these samples were then classified as either mixopositive or mixonegative. What this means is whether we could detect M. xanthus in it or not. And then we also measured or tested for frequency of resistant bacteria in each of these samples. And since I thought, audience, some of you might have not been to ISC, just to advertise the place, it's a very green campus, and also tells you that in a city like Bangalore, it's a little bit of a pristine environment. So our study is sort of not very biased by a lot of anthropocentric effects that you might see. Okay, so this is a simple protocol. Saheli had these soil communities. She essentially plated them. She dissolved them in a favorite buffer of her choice. Took an aliquot of that buffer because idea is that now microbes are sort of now in that buffer. And then plated that on a media that has no antibiotics, and this will give her a proxy for the total number of bacteria that can grow on this in this media. And also she plated the same culture or a same buffer solution in a media that has one specific antibiotic. So this will tell her percentage of bacteria that are resistant to this specific antibiotic in this particular soil sample. Now, of course, these kind of assays are biased because of the type of media you use, antibiotics you use, concentration of antibiotics you use. You just have to believe me, we've taken care of all of those basic sort of controls that one can think of. And this is sort of a snapshot or a overview data that you can get, a bird's eye view of the data set that you can get. So when you check for these multiple different antibiotics, and at that time we had checked only for 6 antibiotics, what we realize is that whenever M. xanthus was present in a given soil sample, that soil sample generally had higher frequency of antibiotic resistant bacteria compared to a soil sample where we did not see or could not detect Myxococcus xanthus. Now, this data clearly shows that presence of M. xanthus is correlated with the abundance of antibiotic-resistant bacteria. I completely agree. What you all might be thinking, this does not show causation. I'm not saying at this stage that M. xanthus is responsible for this change, but there's good reason to test for that possibility. Can we do it? The answer is yes. So how we did this experiment is next we sort of brought these soil.
Sorry, so Mike, can I ask a really quick question?
Yes.
What's the ratio in your samples? What was the ratio of Myxococcus positive to negative in your
Ah,
soil samples?
so I think we got a ratio of about 60 to 40%.
Okay, so fairly balanced.
Yeah, as in we were expecting 40 to 60 other way around.
Yeah, okay.
Yeah, but
Okay.
yeah, as in you can't
Yeah,
control
yeah,
these things.
cool.
Okay. Okay, so the simple experiments, as you all can imagine. So from here on, we sort of moved these communities to lab. As in the primary observation was in the wild communities, but now we sort of picked up some soil, put it in our favorite media. It was a proteinaceous medium, and incubated it along with Myxococcus xanthus for 4 days. And of course, as a control, we had a situation where we did not add M. xanthus to these communities, and again sort of plated them on antibiotic media to check for frequency of resistant microbes. So does introduction of M. xanthus sort of influence frequency of resistant microbes? And this is again one soil sample. We've done these experiments for across different soil samples. And again, we see across these soil samples, whenever we add myxobacteria in soil community, we generally have much higher frequency of resistant bacteria compared to sort of when we don't add Myxococcus xanthus. So then it does associate some causality that M. xanthus is indeed responsible or most likely responsible for higher abundance of resistant bacteria in soil communities also. So this was well and good, but as you can see, this is fairly dirty experiment, right? As in we are doing a 4 day long batch culture, and this is basically telling us, yes, frequency of resistant microbes have increased or not. But we sort of were not careful enough to study it further as to, how much it is increasing, at what time point is it increasing across these 4 days, etc. And therefore, Saheli repeated these experiments. But in this case, she decided to monitor frequency of resistant bacteria every day for these 4 days. And on top of that, we also looked at population of M. xanthus. So yes, we are adding M. xanthus in these complex communities. Essentially, we in the previous data set we looked at what is the frequency of resistant bacteria after 4 days, but we have no idea about the fate of M. xanthus. As in most commonly expected fate is that whenever you add a new member or new microbe in a community as complex as soil, that bacteria would not be able to invade and most likely sort of die out very soon, right? So first question is Mxanthus even surviving in these populations? If yes, how much, and relative frequencies of these bacteria compared to their starting point? That's first. Second, at what time during these 4 days do we actually see increase in frequency of resistant bacteria? So let's first look at what happened to the population of Mxanthus. What we see is that now this sort of is not the greatest representation of this data, and we have enough information and statistics to back this up. But what we clearly see is that from day 0 to day 1, Mxanthus population significantly increases. This is a log scale, by the way. And if you actually look at the ratio of Mxanthus population relative to rest of the microbial community culturable in this particular environment, then in fact that ratio is also leaning towards sort of Mxanthus. That means Mxanthus is indeed sort of invading this community for first day, first 24 hours. After those 24 hours, we basically could not detect Myxococcus xanthus in this population at all. So we have a situation where this predator is surviving actually very well, thriving for first day, and second day onwards, we don't detect these bacteria in any of the soil communities. This is again one representative soil community. They just don't survive in any of the soil communities after 2 days. Okay, now on the same time scale, let's look at what happens to frequency of resistant microbes. And again, the same experiment that I talked about before. And this is how resistant bacterial frequency actually is changing. So what happens is only when Mxanthus is dead, frequency of resistant bacteria is increasing. Which sort of is interesting. This data, of course, was an attempt to try and understand what happens in this community, but has now raised us two questions. One, why is Mxanthus dying? And second, why is frequency of resistant bacteria increasing only after death of myxobacteria? And this is sort of consistent across all soil communities that we've tested. This is sort of a representative graph for one of those communities. Okay, so based on these observations, we sort of generated a conceptual model, and this is how we thought, the underlying mechanism might be. And we said, hey, you first need to have some amount of growth of myxobacteria, because across all of these communities, we see that growth of myxobacteria happening. So their density is increasing. So maybe the density plays a role. I'm not going to talk about this data, but we do know that density plays a role in this system. So therefore, you have to believe me that indeed Mxanthus growth is important. And you have a community where you have resistant bacteria, sensitive bacteria, and Mxanthus. So first step might be growth of Mxanthus to attain a certain density so that we can see this effect. Second is death of Mxanthus, which is exactly what we saw. But here we hypothesize that death of Mxanthus might actually accidentally release some sort of a toxin or antibiotic. So now I've pasted this word antibiotic pretty randomly here. In fact, we know antibiotics are not involved in this interaction that I'm going to show you. But just to give you a flavor of what we thought initially at that stage, that we thought when Mxanthus cells are dying, there's and the environment becomes toxic. And this is again not an outlandish hypothesis, because if you think of other microbes that are known to synthesize antibiotics such as streptomyces, if you kill those cells, environment does become toxic. So we said maybe, when Mxanthus are dying, maybe they're also making environment toxic. And this toxicity, we thought, might then enrich antibiotic resistant bacteria. So in this case, we are saying resistance already existed, and death of Mxanthus is sort of enriching that resistance. So that's our sort of working hypothesis at this stage. Now, of course, to show that this hypothesis is indeed correct, what we have to show is that we can collect environmental factors, that is diffusible factors, and show that those diffusible factors themselves can have this effect that we are seeing. So you don't need Mxanthus, you just need factors that are outside in the environment. And those factors should only be present in the environment when Mxanthus cells are dead, right? So this is exactly what we tested. So we took some spent media and added that to the soil community. That spent media came from either time t0, where we started our experiment, time t24, where Mxanthus was actually thriving, if you remember, at 48 hour, where Mxanthus population died. And if our hypothesis is correct, then supernatant from T0 and T24 should not have any effect, whereas supernatant from 48 hour should actually increase frequency of resistant bacteria. And frankly, we have no clear expectation from 72 hour onwards. But we think it might actually be in this range still. Okay, so when we did this experiment, this is what we saw. So we didn't see any increase or any activity of supernatant that was harvested from first 24 hour. As soon as Mxanthus population dies out, you take supernatant from that culture, add that to soil communities, you have increase in the frequency of resistant bacteria, and somehow that activity stays along. So it seems whatever that ingredient is or that diffusible substance is either fairly stable or sort of is is getting generated throughout this time. Okay, so indeed, first step done. Second, the question we asked was, is it true that it is in fact growth inhibitory? You remember our hypothesis that this must be a toxic substance which is now enriching resistant microbes. So we took the supernatant and asked if the supernatant can have any effect on growth of common soil microbes. And by that, I simply mean the microbes that we had in our minus 80s. And then this is how our data essentially looks like. So on the y-axis, we have relative growth of each of these four different bacterial species in the supernatant relative to without supernatant. So all of my data point are below 1, which means in the presence of supernatant, there's less amount of growth of these bacteria. Of course, you need to control for the dilution effects of supernatant addition, etc. We know that, and we've done those controls. And we consistently see that the supernatant does have a negative effect on growth of most bacterial species, or rather all bacterial species that we've tested. So this supernatant is toxic, is growth inhibitory, and is only active and secreted after the death of myxobacteria. Now, here's another issue with this entire hypothesis. We are adding Mxanthus in a complex soil community, and we are seeing, yes, this supernatant is active and these diffusible substances are present after death of Mxanthus, but there's no evidence that it is Mxanthus that is the source of this active molecule. It might as well be some second or third or fourth species in this complex community. So next we ask, is Mxanthus really the source of this molecules or the supernatant or diffusible factors that are sort of activating or increasing frequency of resistant microbes? And for that, what we did is sort of a sledgehammer approach. We just took a growing culture of Mxanthus and lysed it in the lab and add that lysate of Mxanthus on soil communities and asked, will that have any effect? And indeed, yes. So if you take that nice concentrated lysate, dump it on soil communities, we see that this is not the correct axis, but we see that the frequency of resistant bacteria is significantly higher compared to when we don't add the lysate to it. So indeed, source of these molecules is Myxococcus xanthus. Okay, so now we know our hypothesis works fairly well. Mxanthus's death is releasing something that is coming from Mxanthus, and that is increasing frequency of resistant bacteria. The second question that time curve data brought on was why that Mxanthus was dying, and the question was, why is it dying? We don't know why that Mxanthus population is dying. Our hypothesis is that it's most likely because of starvation, and we have some evidence to that. But we thought, what might be the source of death in natural populations of Mxanthus, right? Because at the end of the day, we started these observations seeing some correlation in the nature. And for that, we sort of have to go back to this life cycle of myxobacteria. So if you remember, I said that these vegetative cells, when they starve, about one or less than one actually percent of these bacteria become spores. What happens to rest of them actually is rest of the population dies out. So there's some form of a programmed cell death in the life cycle. So death is a way of life for these bacterial populations of Myxococcus xanthus. And therefore, we asked, okay, during development, if Mxanthus death occurs, is that death that happens during development, is that enough to have this effect that we are seeing? And because we can induce that development in the lab, so what we can do is we can spot a culture of myxobacteria on a media that has no nutrients. These bacteria quickly start forming fruiting bodies, and during this process, they die themselves. And now what we did is we extracted supernatant or extract from these agar plates where Mxanthus have undergone their own programmed cell death and added that extract to soil communities and asked, is that now enough to generate the response that we are seeing? And the answer is yes. So the the death that occurs during the life cycle of Myxobacteria is enough to have or release these ingredients in an environment that are generating a response where frequency of resistant bacteria are increasing. Okay, so far so good. Now, before I go further, I'll just summarize what I talked about. We started with an observation in the nature, one more picture from ISC, an advertisement. If you want to come, please let me know. I would love to host you. We started with a nice observation in the nature. We showed that it's not just a correlation, there's causality involved. We now know that this sort of conceptual framework does work in this case. And when we talk about source of death of myxobacteria, which is sort of the puzzle that we had, we don't know what is the source of death in our population. Most likely starvation, because it is a batch culture. But in the wild, the same starvation induced response that M. xanthus generates is enough to see or give the signals that we are seeing. Uh, Okay, now the holy grail question, what is in the supernatant, right? As in, what is that active molecule? So before sort of I go further, I'll just tell you, we don't know what it is. We have some idea, and I'll just give you a little bit of brief on what we think these molecules might be. So to characterize these supernatants and figure out what is responsible for this increasing frequency of resistant bacteria, we took the supernatant and fractionated them into two size fractions. So molecules that are larger molecules, we call them, so any molecule that is more than 10 kilodalton, we call them large molecules, and anything that is less than 10 kilodalton, we call them small molecules. As in, this is just an arbitrary scale for these experiments that we used. And we asked, if which of these fractions have an effect that we see. So first, let's see if sort of we have our controls working out again. So we did this experiment for three antibiotics that have three different mechanisms of action. Uh, and when we don't add lysate, frequency of resistant bacteria stays extremely low that we can't even measure. In these particular experiments, when we added lysate, frequency increased massively. Somehow, most of those were resistant. Uh, so yes, this was sort of a sanity check that whenever you add this lysate, we see our primary observation. And let's see what is the effect of these two fractions. And this is what we see. For ampicillin, both of these fractions are active, whereas for gentamicin and tetracycline, both of these fractions independently cannot enrich resistant microbes. And this is this I think this data tells us a lot, right? Because what it tells us is that the mechanism that is enriching resistant bacteria are very different for different antibiotics. For ampicillin, it's a mechanism where both of these fractions are enough to give you the response, whereas for gentamicin, you need both of these combinations together to sort of get the observation that we are seeing. Okay, at this stage, we need to now decide which of these fractions we sort of first characterize, and we decided to first characterize these fractions using their biochemical properties, some old school biochemistry. And the question here was, are these fractions still active after they are treated with these three types of enzymes? Proteases. So if the fraction becomes inactive, that means this is sort of proteinaceous substance. Lipase, that means it was lipid substance. And if the fraction becomes inactive with phosphodiesterase, then it has some sort of a phosphodiester bond, which means it is likely to be some sort of a signaling molecule, right? Now, I'll not bore you with too many of these biochemical graphs, but this is essentially what we see. So top fraction that is larger than 10 kilodalton is sensitive to protein or proteases. Not surprising, these are large molecules. And bottom fraction was sensitive to phosphodiesterase, clearly suggesting that it might be some sort of a signaling molecule that we might be dealing with here. We know a little bit about what those molecules might be, but because we haven't yet tested it enough, I don't want to put my neck on the line right now. But just for information for now, we know biochemical properties of these molecules that are responsible, and that's where sort of the idea of mechanisms involved in these interactions and effect of M. xanthus on natural microbial sort of communities is what we know about. But when you look at this picture, and I want to highlight again that we are looking at increase in the frequency of pre-existing resistance. We are not talking about de novo evolution of resistance at this stage. And this sort of led to next series of question that what is the effect of M. xanthus on de novo evolution of resistance? So if then there's no resistant bacteria in the population at all, does M. xanthus have an effect on that? Uh, and I'll show you some of the very old data, some of the newer stuff actually shedding a lot more light. And I've been begging my students to send me some data. They haven't sent, so I'll not show you the latest stuff, but this is still very fun. So I'll still sort of, put you through the grind and show you this data because I think you'll appreciate it. Okay, so what is the effect of Mixo on frequency of resistant microbes? It's a very simple experiment. You have E. coli that you're culturing over time as a control. In this case, I'm going to show you data for rifampicin. Now, currently, we've done these experiments for a bunch of other antibiotics, and of course, patterns are very different across these antibiotics and across replicates. But this is one thing that I just wanted to show you. I thought this was fun. So this is your control where you're propagating a prey, a possible prey in isolation versus the same prey in presence of myxobacteria and asking whether myxobacteria's presence has an effect on evolution of resistance at all or not. The answer is, of course, in both of these cases, that is prey alone or prey with predator, E. coli, and we are looking at resistance in E. coli, not resistance in predator, resistance in E. coli, that is resistance in prey. In both of these cases, resistance does emerge within first 2-3 transfers, so it's not surprising. Uh, but what I want to highlight here is that when we are looking at resistance, we generally do these experiments where we add media with a given antibiotic and look for growth of microbes, right? And when you do these experiments and control growth of microbes in the presence of a given antibiotic and in the absence of it, you will generally find this pattern. Even if a microbe is resistant to an antibiotic, it tends to grow slower, generally, not always, than in the absence of that antibiotic. So even if it's a resistant microbe, if it's sensitive, of course, it will not grow at all at an inhibitory concentration, right? And therefore, if you take a ratio of growth in these two environments, you're generally likely to see your data point sort of in this bottom half of this graph, closer to 1 or at the bottom half. In these first set of experiments, which were done actually about 4 or 5 years ago, when we did these experiments with rifampicin for E. coli, this is how the data looked like. So E. coli in isolation sort of grows similarly in the presence and absence of antibiotic. This is exactly what traditionally we see. But when these populations and in those particular sets of experiments, when we sort of look for growth of E. coli in the presence of rifampicin versus in the absence, somehow our microbes are growing much better in the presence of antibiotic than in the absence of this antibiotic, which I thought was very interesting because essentially what we are seeing is a situation where antibiotics are sort of promoting growth of bacteria than inhibiting it, and again, we haven't been able to follow up these observations much, but I thought this was sort of an interesting bit to talk about how evolution might function. Uh, so with this, I'll sort of summarize this talk. And what I wanted to highlight is I really like this map that Tom Bokel had sort of mapped out in his paper. And this talks about sort of global south and distribution of resistant bacteria. I think in this case, this graph is made for aminoglycosides. And what essentially how you read this is, there are pockets where sort of 70% of the most commonly used aminoglycosides are most likely to be useless, and he's done that for across different classes of antibiotics. And of course, this is a problem that is sort of, made much worse because of the way we are using antibiotics, especially in most of the global south countries, including India, which is home for me. Uh, what I want to highlight is that I showed you a nice example of a situation where if you already have resistance, microbial interactions would determine how long it will persist. So you remember in the first part of this talk, when I showed you effect of M. xanthus on frequency of resistant bacteria, those that in that environment we didn't add antibiotic at all, right? So if you have resistance, species like xanthus and predators like xanthus might actually be able to maintain it even if we somehow magically manage to remove antibiotics or stop taking antibiotics at all, right? So we need to sort of look at microbial interactions when we are looking at solution to the problem of already spread resistance. That's one. Second, I also showed you an example. Of course, we didn't talk about the mechanism. We don't know all the answers about that example, but I showed you an example where selection pressure that is imposed by predator on a prey leads to evolution of resistance strategies where bacteria ended up growing better in presence of antibiotic, right? Now, essentially what we're talking about is we have a situation currently where antibiotics are ineffective in some cases when the resistance has emerged. But if you have resistance mechanism similar to what sort of we observed at that stage, if you give antibiotic, you're essentially sort of promoting growth of bacteria, possibly making infection even worse. Again, sort of a nice example of how microbial interactions would sort of open up a completely new Pandora's box, which probably we haven't explored yet. Of course, I'm not saying we are not looking at it, but I think in this particular case, it sort of gave us two very interesting observations, which sort of are initially fairly counterintuitive. And with this, I'll stop. And this is my group at the Indian Institute of Science. And this was from our retreat last year. And I've highlighted some of our funders. I've grown to realize that science has become a lot of economics. So major thanks to my funders also. And thank you for your time.